Hello everyone. Now we will see about the gluconeogenesis, its pathway and significance. In earlier class, we have seen that glycogen metabolism, in that glycogenesis and glycogenolysis. Now we will see what is gluconeogenesis. Okay, before going to gluconeogenesis, we will see a brief introduction about the carbohydrate metabolism. Okay, now in carbohydrate metabolism, we have seen that first cycle glycolysis, where glucose is converted into pyruvate and under aerobic condition, then pyruvate converted into lactate under anaerobic condition. Next cycle is TCA cycle, also called as Krebs cycle, where oxidative pathway, in that pyruvate converted to acetyl CoA. That acetyl CoA reacts with oxaloacetate to form citrate. To form citrate. That citrate undergo again series of reaction to form a oxaloacetate. This will be again recycling of uh, what we can say, formation of citrate and oxaloacetate, so that huge amount of energy will be produced through the TCA cycle. Okay, next is HMP pathway, where is production of pentose, and after that we have seen lastly glycogen metabolism, in that glycogenesis and glycogenolysis. Glycogenesis is synthesis of glycogen, whereas glycogenolysis is breakdown of glycogen. Okay, next is gluconeogenesis. Now gluconeogenesis contains three words: gluconeogenesis. Gluco means glucose, neo means new, and genesis means synthesis. So synthesis of glucose from the new compounds. We know that when we will take the food in the form of carbohydrate, it will get converted into glucose in body to supply the energy. But certain non-carbohydrate molecule like pyruvate, amino acid, and fats will also get converted into glucose. This conversion of non-carbohydrate molecule into glucose, that process is called as gluconeogenesis. Synthesis of glucose from new rather than or other than carbohydrate uh, compounds. Okay, now you know that blood glucose level, normal blood glucose level is 100 to 200 mg per deciliter or 100 ml. Okay, when there will be excess of glucose, that glucose will be stored in the form of glycogen in liver, and whenever there will be a fasting or exercise, there will be a decrease in blood glucose level, because that glycogen from the storage of liver will get breaks down to glucose to supply the energy and to maintain the blood glucose level. Whenever there is a continuous utilization of glucose, there will be a continuous breakdown of glycogen. That continuous breakdown of glycogen into glucose, then that glucose continuous utilization for energy, that will lead to the decrease in the blood glucose level. Are you getting? कंटिन्यूस यहाँ पे जब ग्लूकोज का बॉडी में यूटिलाइजेशन होता है, तब उसका ब्लड ग्लूकोज लेवल कम हो जाता है. एट दैट कंडीशन व्हाट हैपन्स, नाउ सर्टन पार्ट इन द बॉडी like brain, which require continuous supply of glucose for their functioning. Okay, there will be a continuous supply of or continuous requirement of glucose for brain for their functioning. Now, under these circumstances, when there will be a low blood glucose level, okay, certain non-carbohydrate molecules like amino acid, fat, and pyruvate get converted into glucose to fulfill the blood glucose level or to fulfill the body glucose requirement. Okay, these metabolic reactions involved gluconeogenesis, means the reaction involved in the gluconeogenesis are glycolysis, Krebs cycle, and some other reactions. Means whatever reactions involved in gluconeogenesis, means synthesis of glucose from non-carbohydrate, those reactions are from the glycolysis, Krebs cycle, and some other reactions. So if you know the glycolysis, Krebs cycles, or if you are very good with these two cycles, there is nothing in gluconeogenesis. Okay, now what will happen in gluconeogenesis? First we will go through glycolysis in brief. You know that glycolysis is a breakdown of glucose 6 carbon atom into pyruvate. Two pyruvate molecule of 3 carbon atom will produce. There will be a series of reaction, glucose to glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bisphosphate. Then that will be converted into dihydroxyacetone phosphate, glycerol diet 3 phosphate. This glycerol diet 3 phosphate converted into 1,3 bisphosphoglycerate, 3 phosphoglycerate, 2 phosphoglycerate, phosphoenol pyruvate, and finally pyruvate. This is under aerobic condition. Under anaerobic condition, it will get converted into lactate. Okay, totally 10 step of reactions glycolysis is. Out of 10, 3 reactions are irreversible. First one is glucose to glucose 6 phosphate is irreversible, fructose 6 phosphate to fructose 1,6 bisphosphate, and phosphoenol pyruvate to pyruvate. 1, 2, 3 are irreversible, otherwise all are reversible reaction. So whenever there is a deficiency of glucose in blood, especially in fasting, this irreversible reaction upregulated. Means whenever there is a blood decrease in blood glucose level in the body, these इरिवर्सिबल रिएक्शन जो ये 3 है वो अप रेगुलेटेड मीन्स रिवर्स हो जाती है to fulfill the requirement of glucose. Usually these are means once glucose is converted into pyruvate, it will not get converted back to glucose, but in certain conditions like fasting or exercise or starvation, these 3 reactions will get reversible. But these 3 reactions will not contain same enzyme as in the glycolysis. Otherwise, all these different 7 reactions will be same, and those contain same enzymes as in glycolysis, whereas these 3 steps will not contain same enzyme as that in glycolysis in gluconeogenesis. So you have to remember these 3 reactions in gluconeogenesis. Okay, in glycolysis you see here a hexokinase enzyme is used, here phosphofructokinase enzyme is used, and here pyruvate kinase enzyme is used. But these 3 enzymes will not be there in gluconeogenesis. There will be a different 3 enzymes to reverse this reaction. So pyruvate converted to glucose. Okay, now one more TCA cycle. We have seen that in TCA cycle pyruvate converted to acetyl-CoA. Acetyl-CoA reacts with oxaloacetate to form citrate, then isocitrate, then oxalosuccinate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate again. Now what happened? This oxaloacetate will get converted into phosphoenol pyruvate. Okay, this phosphoenol pyruvate is seen here. After pyruvate, phosphoenol pyruvate will come, and again this after conversion of oxaloacetate to phosphoenol pyruvate, this phosphoenol pyruvate will get converted to glucose through reversal of glycolysis. Okay, then one more is the propionate. Now propionate is a non-carbohydrate compound obtained from the fatty acid oxidation. This propionate or propanyl-CoA will get converted into succinyl-CoA. This succinyl-CoA is an intermediate of TCA cycle. Again it will get converted to oxaloacetate. That oxaloacetate again converted into phosphoenol pyruvate, and that phosphoenol pyruvate again through reversal of glycolysis converted to glucose molecule. Simple steps are there. Just you have to remember the glycolysis process very well. Okay, once if you are pakka with the glycolysis, there is nothing in the gluconeogenesis. Now that one was the introductory part I have told you. Now coming to notes and cycle. Okay, gluconeogenesis pathway and significance. Maybe for 5 marks or 10 marks they will ask. Okay. Now definition of gluconeogenesis is the synthesis of glucose from non-carbohydrate compound is known as gluconeogenesis. Already we seen the major substrate or precursor for gluconeogenesis are lactate, pyruvate, glucogenic amino acid, propionate, and glycerol. Means whatever starting material required for the gluconeogenesis are lactate, pyruvate, glucogenic amino acid, propionate, and glycerol. From this non-carbohydrate compound, glucose is formed. Location mainly cytosol and some precursor in mitochondria. Now, what are those precursors in mitochondria? We will see later. Now, it will mainly occurs in liver and some extent in kidney and matrix. Gluconeogenesis mainly occurs in liver and some extent in kidney and matrix. Kidney matrix. Now, reactions of gluconeogenesis. There are totally 5 main reactions through gluconeogenesis occurs means formation of glucose. First from pyruvate, second from lactate, third from propionate, that is propionyl CoA, then fourth one is from glycerol, and fifth one is glucogenic amino acid. Now first we will see from pyruvate. Now from pyruvate it is closely resembles to reversible pathway of glycolysis. As earlier told, from pyruvate how glucose will form, it is just like a reversal of glycolysis only. Only there are 3 irreversible reactions in glycolysis out of 10, and 7 are common in both glycolysis and gluconeogenesis. आपको क्या करना है, ग्लाइकोलाइसिस के जो स्टेप है वो उल्टा लिखते जाना है. उसमें 3 जो इरिवर्सिबल रिएक्शंस है उसके एंजाइम्स चेंज हो जाता है. ओके, दैट वी हैव टू सी हियर हाउ pyruvate converted into glucose. Okay, now in that first step is conversion of pyruvate to phosphoenol pyruvate. This takes place in 2 steps. I will directly go to cycle now. How pyruvate converted to phosphoenol pyruvate. Now this is a pyruvate molecule. You know the final product of glycolysis. This pyruvate, with the help of enzyme pyruvate carboxylase, will get converted into oxaloacetate. Now pyruvate usually present in the cytosol. You know, glycolysis will happen in cytosol. This pyruvate will enters to mitochondria, where it will get carboxylase, means addition of carbon dioxide will get place. In the presence of pyruvate carboxylase, you can see C double bond O CH2, C double bond O COO COO. Okay, so there will be addition of carbon dioxide in pyruvate to form oxaloacetate. But here one thing happens, this oxaloacetate impermeable to semi-permeable membrane, okay, or mitochondrial membrane. It will not come back to the cytosol oxaloacetate because the remaining enzymes are present in the cytosol. Once pyruvate enters to mitochondria, it will get converted to oxaloacetate, but this oxaloacetate will not come back to cytosol. For that oxaloacetate again converted into malate in the presence of enzyme malate dehydrogenase in mitochondria. Now this malate is permeable to semi-permeable membrane of mitochondria, and this malate is come back to cytosol. Okay, now in cytosol this malate again converted into oxaloacetate. ये इम्परमिएबल रहता है, इसके लिए ये इसका पहला कन्वर्जन मैलेट में होता है। मैलेट परमिएबल होता है, वो साइटोसॉल में आ जाता है, फिर इसका ऑक्सैलोएसिटेट में कन्वर्जन होता है। बिकॉज फ्रॉम ऑक्सैलोएसिटेट ओनली phosphoenol pyruvate will form. Now from oxaloacetate phosphoenol pyruvate form through phosphoenol pyruvate kinase enzyme. Here one molecule of GTP is converted into GDP, and also one carbon dioxide molecule will be removed out. You can see this CO is removed out, CH2 double bond will form there. Then at second carbon O phosphate group will be attached from one GTP to conversion to GDP. Now you know that last step of glycolysis is conversion of phosphoenol pyruvate to pyruvate. So here pyruvate to phosphoenol pyruvate through 2 steps. Now this is actually first step of gluconeogenesis. Now second step is second step is phosphoenol pyruvate get converted into 2-phosphoglycerate. Third is 2-phosphoglycerate to 3-phosphoglycerate, 3-phosphoglycerate to 1,3-phosphoglycerate, 1,3-phosphoglycerate to glyceraldehyde and dihydroxyacetone phosphate, and dihydroxyacetone phosphate to fructose 1,6-phosphate. Means from step 2 to step 6 are just a reversal of glycolysis. There will not be any changes in enzyme and product. Okay, you can see in glycolysis fructose 1,6-bisphosphate converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then it will get converted into 1,3-bisphosphoglycerate, then 3-phosphoglycerate, then 2-phosphoglycerate. But here reversal will happen. 2-phosphoglycerate, phosphoenol pyruvate converted to 2-phosphoglycerate, then 3-phosphoglycerate, then 1,3-bisphosphoglycerate, then glyceraldehyde and dihydroxyacetone phosphate, that will be converted to fructose 1,6-bisphosphate. Just a reversal 2 to 6 step. आफ्टर दिस फर्स्ट स्टेप 2 to 6 आपको जस्ट ग्लाइकोलाइसिस के रिवर्स स्टेप लिखना है. ओके, आफ्टर 6 स्टेप fructose 1,6-phosphate bisphosphate. Now this one already told, this one also irreversible reaction. There are total 3 irreversible reaction in the glycolysis process. This fructose 1,6-bisphosphate from this one phosphate molecule will be removed out with the help of enzyme fructose 1,6-bisphosphatase. ओके, एक फॉस्फेट मोलिक्यूल निकल गया है बिस्फॉस्फेट में से, ओनली सिंगल फॉस्फेट मोलिक्यूल रिमेन्ड. नाउ दिस इज फ्रुक्टोज 6-phosphate. अगेन similar reaction from the glycolysis. Phosphohexokinase will convert fructose 6-phosphate to glucose 6-phosphate. Just only change of functional group here. In glycolysis also we have seen that from glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate. Here also from fructose 6-phosphate to glucose 6-phosphate. Now last step is glucose 6-phosphate to glucose. This is also irreversible in glycolysis, where hexokinase enzyme was present. That enzyme will not be here. Here glucose 6-phosphatase. Now wherever phosphatase enzyme will come, that enzyme's main function is removal of phosphate group, phosphatase. Whenever isomerase will come, there will be a change in the functional group. Whenever there is a epimerase enzyme will be there, there will be a change in the what we can say, change in the position of the atom in the molecule. Okay, so final step is from glucose 6-phosphate phosphate to glucose. So we have seen here how pyruvate molecule is converted into glucose. It is very simple. Just you have to get pakka with the glycolysis, then gluconeogenesis is very easy. If you know the glycolysis very well, only 3 steps are irreversible. At that 3 steps you have to add different enzymes, and you have to write same uh, reactions reversibly, like from pyruvate to phosphoenol pyruvate. Here some matter will come, then that phosphoenol pyruvate to up to fructose 1,6 bisphosphate, same glycolysis reactions will come. Now here also same similar reactions are there, fructose 1,6 bisphosphate to fructose 6 phosphate, only enzymes. Here phosphatase enzymes will come, here glucose 6 phosphatase enzymes will come, whereas we have seen uh, phosphofructokinase will come here, and here hexokinase will come, or glucokinase in glycolysis. Okay, this is about how pyruvate will get converted into glucose, and it is explained in notes also here, step by step. Okay, now next, how gluconeogenesis from lactate. Now under anaerobic condition, pyruvate is converted to lactate by lactate dehydrogenase. Lactate Now lactate is majorly present in skeletal muscle. Next it is carried from skeletal muscle to blood, where it is oxidized to pyruvate. जो भी लैक्टेट फॉर्म होता है वो स्केलेटल मसल में रहता है, उसको बाद में ब्लड में ट्रांसफ़र होता है, और वहाँ पे उसका ऑक्सीडाइजेशन होके वो पायरुवेट में कन्वर्ट होता है. देन दैट पायरुवेट अगेन कन्वर्टेड टू ग्लूकोज थ्रू ग्लूकोनेओजेनेसिस. सिंपल. अगर आपको एग्जाम में पूछा है राइट अ नोट ऑन कोरिस साइकिल, then you have to start with lactate. Okay, gluconeogenesis from lactate. Now gluconeogenesis from lactate contains only one extra step than the pyruvate. Here lactate will get converted into pyruvate through enzyme lactate dehydrogenase. Then that pyruvate will get converted into glucose. Whatever earlier we have seen, that you have to write completely. Suppose in exam gluconeogenesis, write a note on gluconeogenesis. They told you have to write first gluconeogenesis from pyruvate in means you have to write in depth that one, but gluconeogenesis from lactate you can write, this one will be enough. Okay, now next, gluconeogenesis from amino acid. Okay, certain glucogenic amino acids except leucine and lysine, कुछ ऐसे अमीनो एसिड्स ग्लूकोजेनिक अमीनो एसिड बोलते हैं, they will readily goes in pyruvate form. Okay, again that pyruvate will get converted into glucose through gluconeogenesis. Simple. Okay, now next, gluconeogenesis from glycerol. Now glycerol is liberated in mostly in adipose tissue by hydrolysis. The enzyme glycerokinase, okay, in liver converted it to glycerol 3 phosphate. Then glycerol 3 phosphate converted into dihydroxyacetone phosphate by enzyme glycerol 3 phosphate dehydrogenase. Now whatever glycerol will be there mostly present in or liberated in adipose tissue, that glycerol is converted into glycerol 3 phosphate by glycerokinase. Okay, glycerol also considered as a lipid molecule. Okay, non-carbohydrate molecule. From that how glucose will form, first glycerol will be converted into glycerol 3 phosphate with the help of glycerokinase enzyme. You can see one phosphate group is attached here. Okay, now next, glycerol 3 phosphate will be converted into dihydroxyacetone phosphate with the help of enzyme glycerol 3 phosphate dehydrogenase. Now you know that this dihydroxyacetone phosphate is intermediate of glycolysis, so it will get involved in the reversal of glycolysis to produce the glucose. Simple. Okay, now next is gluconeogenesis from propionate. How gluconeogenesis from propionate. Propionate obtained from the oxidation of odd chain fatty acid. Now, odd chain fatty acid, even chain fatty acid, what are the saturated, unsaturated fatty acid will comes in next uh, topic, that is lipid metabolism. I will explain there, but just you remember oxidation of odd chain fatty acid will yields propionyl CoA. Now this propionyl CoA in the presence of enzyme propionyl carboxylase gets converted into succinyl CoA, and you know that succinyl CoA is intermediate product of Krebs cycle. Okay, now this succinyl CoA is converted into oxaloacetate. This oxaloacetate will convert it into pyruvate, and this pyruvate again converted into glucose. So every gluconeogenesis process, you can see that intermediates what are there in glycolysis or Krebs cycles are involved. Okay, so if you know glycolysis and Krebs cycle very well, this is a very simple cycle to understand. Okay, after this, next is significance of gluconeogenesis. What are the significance of gluconeogenesis? First one is reversal of glycolysis steps where pyruvate converted to glucose. As earlier I told, it is a reversible process of glycolysis steps where pyruvate converted to glucose. Second, it maintains the blood glucose level during starvation, fasting, and prolonged exercise. Okay, next, brain, CNS, that is central nervous system, mainly dependent on glucose for continuous supply of energy. So in fasting they are mainly depend on the gluconeogenesis. Okay, this all the points are self-explanatory. I will not go in depth. Certain metabolites produced in the tissue accumulate in the blood. ओके, कुछ मेटाबोलाइट्स होते हैं जो अक्यूमुलेट होते हैं ब्लड में, लाइक लैक्टेट, पायरुवेट, ग्लिसरॉल, प्रोपियोनेट, जो हमने प्रिकर्सर देखे ग्लूकोनेओजेनेसिस में, दोज आर इफेक्टिवली क्लियर्ड बाय ग्लूकोन. इनको ग्लूकोज में कन्वर्ट किया जाता है एंड दे विल बी इफेक्टिवली रिमूव्ड फ्रॉम द ब्लड टिश्यू. ओके, now next it will regulate acid base balance, also gluconeogenesis. Next, it removes lactate from skeletal muscles and RBCs, and lastly it is a de novo synthesis of glucose. It is also called a de novo synthesis of glucose. Suppose in exam de novo, write a note on de novo synthesis of glucose, you have to write gluconeogenesis only. Okay, so I hope this will be clear to you. If any doubt, please ask. Thank you.